without further ado, it is my pleasure to introduce our speaker today. Dr. Marta Veses Garcia is our head of assay development, and she is going to be talking about rapid sterility testing for cell and gene therapy products. Over to you, Marta.
Thank you, Ben. Hi, everybody. Thank you for joining us today. I'm going to give you an overview of our solution for rapid sterility testing for cell therapy and gene therapy products. They is based on our biocalorimeter, the calScreener Plus. So I'm Marta, I'm a microbiologist. I've been working at Symcel for a little bit over 6 years now. I'll start with a brief introduction to the company, and then I will dig into the more technical aspects of what calorimetry is and how do we use it for sterility testing of mainly cell therapy products, how the data looks like, how do we analyze it, and how far we are in the process now of validation. So the last few slides will cover a little bit of where we are in the validation journey. Change. Yeah, so the same, we are Symcel, a company founded and located mainly in Stockholm, Sweden. So now we have expanded into the US, a little bit of Denmark, and we have representation as well in China. When I joined over 6 years ago, I was the third person employee, and I was an application scientist. And as we've been expanding, developing, and growing, now we are over 40 people of many different nationalities. It's been a very exciting journey, and we have found a very, very good niche for our technology, for biocalorimetry, in microbial detection. Not just in sterility testing, that is what we're dealing with today, but also in the clinical world, right? We also have a clinical program to develop diagnostic device for prosthetic and joint infections. So what we do is we manufacture a biocalorimeter, the calScreener Plus. Well, the calScreener is the original instrument that is dedicated for mainly research. And then we have updated the instrument to fit the sterility testing market, and we call it the calScreener Plus system. Maybe you're already familiar with microcalorimetry, also called isothermal microcalorimetry, because it's mentioned in both the US and the European pharmacopoeias as one of the rapid methods for microbial detection and sterility testing. Although it's not a regulated and approved method yet, we hope to change that very soon. And in essence, microcalorimetry measures heat from chemical reactions. Any chemical reaction, endothermic, exothermic, produces absorbed heat, and that's what microcalorimetry measures. And it is specifically isothermal because the systems get a very stable, unique U1 temperature. When we apply isothermal microcalorimetry to the biological systems like we do here for sterility testing, then we call it biocalorimetry. Since all live organisms, all cells that are alive produce, they have a metabolic activity, they're metabolically active, and metabolism is the chemical reactions, right? So all metabolic processes produce heat. And that heat produces directly proportional to the metabolic rate. So the more metabolically active an organism is, the more heat it produces. And we use that to very rapidly detect any increase of metabolic activity in a sample. It is it's non-discriminatory. So we don't measure specific metabolic pathways. We don't measure just respiration, just fermentation, just growth. We measure the net output of metabolic reactions. And in that sense, it's also non-discriminatory between eukaryotic and prokaryotic cells. But I will show you later on how do we distinguish between eukaryotics and prokaryotic, because that's a question that comes very often. We are developing this system specifically for gene and cell therapy products. Many of them are T-cell based. How do we distinguish between a eukaryotic and prokaryotic or fungal cells? And I will show you that in later slides. But a little bit of a schematic here on how the technology actually works in our biocalorimeter. We have these small glass vials that serves as an incubator. Basically, each individual vial is your own cell incubator. You put your sample there together with bacterial growth media. And once it goes inside the calorimeter, each vial sits in one individual heat detector. They are also they're enclosed in a loading plate that keeps them isolated from each other. So the heat, if there is some heat dissipated, it doesn't contaminate each other's samples. Then the sensors sit on a heat sink, which is a block of 1 kilo of aluminum. And there are several physical processes that direct the heat towards the heat sink and through the heat detector. So the moment there is metabolic activity in the vials from live microbes, the heat detector detects that metabolic activity. This is one reason why it's very, very important that the system is isothermal and is very, very stable temperatures, because the delta, very minute deltas in temperature between the sample and the system is what it's measured and then converted into an electrical signal by a Peltier element. So at the end, what we see is a measure of power, microwatts of joule per second. This is the actual biocalorimeter, which is a benchtop instrument. For sterility testing, we work with 2 units, one at each temperature, to follow the compendial recommendations. So the temperature range that we work, the biocalorimeters can work with is 20 to 40 degrees, and we are optimizing, validating the sterility testing assay at 25 and 35. That's on the high end of the temperature recommendations by the pharmacopoeia, 20 to 25, 30 to 35. But one particular aspect of this instrument is that they have a very, very, very tight temperature control. So we can confidently set them at the higher end, 35 and 25. That is what we found to be the most optimal for a fast detection of most microorganisms that we work with. And we are sure that we're not going to go over and we can accept the results. But as I say, each unit is set at one temperature. So to run the compendial assay, you will need to have 2 units. Then this is the plate where you put your samples, and this is the disposable glass vials with print cap, where you would inject the media, your samples, place them in the plates, and then in the calorimeter they go. There's a position for, it is a 48 well plate, but 16, the first row and the bottom row are thermodynamic references. So they are needed for the stability of the signal and to filter any noise that might come from the environment. So there's a position for 32 samples, parallel testing. And the calorimetry is controlled by a software that is a 21 CFR Part 11 compliant, and also has we have implemented a machine learning detection algorithm for unbiased detection of microbial activity. So once you put the plate in, you don't have to do anything else. Start the software, start collecting data, and the software, the moment detects microbial activity, is going to flag that sample as positive. Also, the measurement is continuous and live, so there is no interruption in the signal. A little bit go over the workflow, which is really very simple. Is taking your glass vials and not pipetting, but injecting media and sample. Once you do that, you crimp them again to seal them completely and put them on the glass in the loading plate and into the calorimeter. The loading into the calorimeter is manual, and because you need to open this arm here to put the plate in and push it in, there is a disturbance of the temperature inside. And as I say, it's a very sensitive instrument, but it's very important that it's kept at the same temperature in isothermal conditions. So there is a recovery time for that temperature to equilibrate once you open and disturb the temperature inside. That takes about between 30 and 40 minutes, not long. And after that, you start collecting your software, the data, and just wait until the run is over or until you have a positive. This is the graphical output of the data that is collected in the calorimeter. So this is called a thermogram. When we refer to biological system, we also call it the metabolic fingerprint of metabolic profile. The axis, we have the heat flow in microwatts over time. And this is the thermogram for Staph aureus in TSV. So you can see a rapid increase of metabolic activity, then there are some inflection points, and until we reach a peak, and then it's the decrease, kind of stabilization, and then it goes down again. We have characterized and matched the metabolic activity, especially at the beginning of the curve for many organisms, to match it with the exponential growth phase. And you can be quite sure that this, at least until the first peak, usually matches exponential growth phase. So the energy they are producing at the beginning of the curve up to the peak is usually associated with biomass increase. And what else I wanted to tell you here? Oh, the time to detection, the time that we start to see an activity, it's directly proportional to how many cells do you have initially in your sample. So the more cells you have, the faster you will detect activity and so on. A very useful and particular characteristic of biocalorimetry is that each metabolic profile that we have seen from different species is unique. So depending on the culture conditions, each species has a different thermogram. Here I'm showing you the compendial organism thermograms at 35 in FTM. You can see how different they are, with Pseudomonas having a very quick outburst of metabolism of heat production, and then quickly coming down. With something like Clostridium or Staph aureus, they have a much lower peaks, but they continue producing energy for a longer period of time. And then in TSV at 25 degrees, we have the 3 environmental isolates. And now the scale is much lower, but I really wanted you to appreciate all the inflection points that we have in the curve and how this can be used for species identification. This is something that we have already implemented for a clinical development program. And it's currently not a feature of the calScreener Plus system for sterility, but it could be included in future releases if this is of interest. Now, for identifications based on the thermogram, you will have to have some of the, maybe not all the thermogram, you don't have to wait 48 hours to have a complete profile and be able to identify the species, but a little bit more than just the beginning, right? But for detection, we have implemented a machine learning algorithm and trained it to recognize very fast inflection points in the metabolic activity. So I'm going to zoom in now in Candida and Bacillus to show you that actually this increase of activity very early on is what the algorithm detects and flags as a positive sample. Before that, we either have a plateau or a negative slope. So a curve that always goes constantly downwards. The moment there is an inflection point and the activity shoots up, there is a positive slope. The algorithm can detect that and be confident there is a positive microbial activity with a delta of 1 microwatt. This is something that we have implemented for the last 2 years and now works really, really well. And it can give us a very, very fast time to detection. And linking to that, this is the growth promotion time to detection. So the 6 compendial organisms that we have here in the recommended pharmacopoeia incubation conditions. So we have Clostridium, Pseudomonas, and Staph aureus, and you see the very fast detection, very fast growing organisms within 18 hours. All of them have, and all the data that I'm going to show you is starting with less than 5 CFUs in the vial. So we inoculate the vials directly with less than 5 CFUs. We mostly work with bio balls, certified material from bioMérieux. This is very easy to control. And if they don't have the material doesn't exist for bio balls, then we use microbiologic RQ shots. But it's always less than 5 CFUs. So yeah, fast growing organisms within 24 hours, within 18 hours, we can have a very fast detection. Environmental organisms, these are the pharmacopoeia recommended conditions, TSV at 25 degrees. And you see that within 2 days, we can detect them all. But actually, I also want to show you the conditions of FTM at 35, because in our experience, running these organisms is always much faster detection. And in some cases, like Bacillus, it's a dramatic decrease of time to activity, right? We have a little bit over 24 hours at 25 degrees and less than 12. I think it's around 8 hours if we put them on FTM at 35. We have done extensive screening of these organisms at different temperatures and in different media, and this is a temperature effect. They're always faster, closer to 37, which is their optimal, than they are at 25. But since these are the recommended, the compendial conditions, I need to show you this. We have also expanded, not only have data for the compendial organisms, but also with more, or especially environmental strains. We have as well strep pyogenes, fast grower, less than 12 hours detection, and also C. acnes that always catches people's attention. We detect within 60 hours, so less than 3 days at less than 5 CFUs in the vial. We have a very consistent detection of C. acnes. And then environmental organisms, and here we have 3 molds and Micrococcus luteus, then there is detection within 48 hours. So now leaving growth promotion and focusing on the sterility assays directly on product, we follow a direct inoculation assay where we just mix the product directly in the vials with media. Typically, we have been testing 1 milliliter samples that then we have to split in 2, 500 goes into TSV at 25, 500 in FDM at 35. And we use 2 vials per condition because we do a 1-to-1 dilution of product with growth media. I will show you later in a few slides how we got to that dilution. That is the one that we found that works best for detection and overcoming potential inhibitions in the sample. Yeah, and this is how the data looks when you have an actual cell in product in the sample and the background that comes with it. So we have in gray the media only, FDM only, no product, no bacteria, and then you see there is already a background reaction. This is because media, we also measure chemical reactions, and FDM in particular that has sodium thioglycolate is reacting with oxygen and most likely is giving us an exothermic reaction. But this is always a negative slope. It's always a decreasing signal. And it's the same for T cells. In this case, we're using Jurkat as a Jurkat cell culture. And we do have a background signal, but it's always a downward slope. And the algorithm is trained to recognize that downward slope and then recognize when the inflection point happens and the positive slope comes from microbial detection. This is always the same. We never get a false positive when there's only Jurkat cells or only media. Yeah, and then I have the other example. There is the acnes that has a longer detection time, so you can see much better how there's always downward negative slope that reaches, and it takes about 72 hours for all the heat to dissipate, and then we have a very clear positive C. acnes detection. And this is the time to detection that we have calculated using the Jurkat cell culture. Is very similar to growth promotion. With the fast growers experiments, no inhibition whatsoever. This within 18 hours we can have a detection. The only one that is slightly delayed is Brasiliensis, but it's also within 48 hours detection, and against the acnes is within 3 days. So the Jurkat cells do not have a significant impact in time to detection for these organisms. And I mentioned before that we do a 1-in-1 dilution product to media, and that is initially we wanted to use as much product as possible in the vial, so we were concentrating the media and adding between 350 and 400 microliters of product. We very quickly realized when we started to run customer samples, then if you use the cryopreservant, DMSO being the most common one, this is severely impact detection and time to detection because it is a microbial inhibitor. So in the most drastic cases, we cannot detect the organism, and other times we experience a very delayed time to detection. Here you have Pseudomonas that usually takes 8 to 9 hours, and here we are over 2 days. And it also pushes C. acnes and Candida albicans over the 3-day threshold that we have. So that's when we started to optimize the method and find the dilution that worked to remove this inhibition. And luckily, just a 1-to-1 dilution. So decreasing the DMSO concentration from 10 to around 5%, already restored both detection but also fast detection. So you see very quickly we go back to less than 24 hours for Pseudomonas, less than 3 days for Clostridioum, Cutibacterium, and 48 hours for Candida. So we were quite happy to see that a very simple solution will give us back very good results in time to detection. And to wrap this part up, I'm going to show you a customer case from Smart Immune, a French company. They very kindly has let us share some of the data from the feasibility project that we run with them. So they have several therapy products, but one of them, Smart 101, the most clinically advanced that they have, we've been testing last year. And they do have a DMSO matrix, and it's one of the ones optimizations that we did. And we were quite happy to see that we got very good results and timing for them as well. So this is as well with a 1-to-1 product media ratio and starting with less than 5 CFUs spiking. You see the fast growers, we all have detection within 18 hours, very fast. Candida on the first 48 hours, and then Brasiliensis, which is one of the organisms that is heavily inhibited by DMSO, we could recover and detect within 72 hours, as well as the others. So we were very happy to see that all of the assay optimization that we have done works for real customer samples. So up to here is the data, the technical data that I wanted to show you, and hopefully I have given you a good overview of the calScreener Plus system that uses biocalorimetry for sterility testing assays. The Assay calorimetry is recognized in both US and EU pharmacopoeias as a fast microbial detection method. And we have developed a fast phenotypic assay that can detect microbial activity of all of the organisms that I have shown you within 3 days, regardless of product or incubation conditions. There is no interference from eukaryotic of media background, although there is a media in a eukaryotic cell background. That is not a problem for the software to detect a positive signal from microbial presence. And this is an objective, automated, yes or no result. So no one has to go going into your data and looking at, is this an increase, is not, the software does that for you. So now just a couple of slides on where we are in the regulatory pathway. We are following of course, the US and the EU pharmacopoeias to validate the assay, and we're following the recommendations for a qualitative assay. Although microcalorimetry can be semi-quantitative, we are focusing for now in a qualitative yes or no answer. So sensitivity, specificity, roughness, and robustness, and equivalency. The sensitivity, the limit of detection studies are ongoing. We're following the most probable number method. We do know, since all of our assays are below 5 CFUs, that we will have a low limit of detection, but of course we need to provide the statistically relevant numbers. Specificity, we have a lot more experience with, since mainly for a clinical development program, we have tested over 60 different species related to the clinical strains, over 500 clinical strains of those 60 species, and also other environmental isolates from research projects. And we are continuously expanding with relevant organisms for sterility testing. And we have done some work already as well in roughness and robustness, and we see that for operators, de facto operators, instruments, and media lots, that we have a very rough system. Equivalency against compendial methods is also ongoing. And we aim to have all of these finished by September, because that's when we plan the launch of the calScreener Plus system, 2024, so not that long to go. And hopefully start validation journeys with some customers over next year. We also have planned a meeting with the Advanced Technologies team at the Center for Biologics in the US with the aim of discussing the primary validation package, but also to submit a Type 5 master file in 2025 for the calScreener Plus system. I think I spoke a bit faster than I thought I would go into, as it usually happens, but I'll be happy to take any questions if you have them. Or you feel free to contact me at any point with information or questions about the data.
Thank you very, very much indeed, Marta, and thank you everybody for listening. I know some of you had to dash off, so to all of you I say thank you. The recording will be available shortly and you'll get it in your email. As Marta said, please feel free to reach out to us with any questions after the event. Share the recording. We're very, very happy to discuss these things. So let's get into some questions. We've already had some coming in throughout the webinar. Please feel free to ask more in the chat or the Q&A. I'll go through them now in order. So basically, one of the first questions we had, and I guess this has been answered a little bit, but perhaps it's worth going through again, is how long is the run? And I guess that depends exactly on what we're looking for. So perhaps you could just clarify maybe how long you need to run the assay for, and perhaps how long might you need to run it for if we were wanting to do something like ID?
Yes, so for detection, that is something that has to be empirically defined for each product. Of course, we need to optimize each product individually. That's something that we have learned over the last year. We aim for 3 or 3.5 days run. So far, I think 3 days will cover most of the organisms. We haven't found any that takes longer than 3 days with an optimized protocol. But of course, that has to be determined case by case, product by product. And for identification, it's I'm not entirely sure, but this it will depend on how many organisms are in the database and how much portion of the thermogram you need. It could be 6 additional hours today, or it could be half a day more if you want identification. Great. But that we need to optimize as well.
So you've basically covered that, I think. And I know I said I'd go in order, but I've seen some questions coming at the end related to time. So can we also claim identification in 3 days? I guess you basically just answered that, but explicitly.
For late-growing organisms like Cyagnus or Aspergillus, it's likely going to take a little bit of over 3 days, but not much more. For most of the organisms that grow within 48 hours, then yes.
Cool. And also a time-related question. So we've charted the time in these graphs now to get a positive. So how long do we need to confirm a negative result?
For the negative? Yeah, that's what the part that needs this to be defined for each product as well. So you need to pick when there are recommendations, especially in USP 72, and there's a formula that you should follow to add time to your, based on your slower organism. So in our case, would be Cyagnus, and then you add some extra time. So probably around 3.5 days, maximum 4 days for a negative result. But this again needs to be calculated individually. Great.
So we've also shown different species at 25 and 35 degrees. Are the thermograms the same for each species at these different temperatures?
You know, I saw that question might come and I didn't have the backup slide for it. If you have the same medium, say TSV at 25 and 35, the thermograms are very similar. The nutrients are the same. What changes is the slopes. They have usually a slower slope at 25. But the inflection points, it's quite far. So for example, if you run Pseudomonas at 25 degrees, the shape would be more or less the same. The peak might be lower, and then the rate, instead of being such a steep slope, it will go a little bit lower, slower. But in essence, the thermograms are the same, as long as the media is the same.
Great. Have we ever tried samples with antibiotics, as some products do contain residual antibiotics?
Yes. Not for sterility. We have run clinical tissue samples that potentially contain antibiotics for a clinical development program. But not for sterility. I imagine it will work a little bit like the MSO. As long as you dilute them a little bit, then you will detect them, but it will depend on the residual concentration of antibiotic.
Understood. Here a question about the limit of detection in microwatts for the calScreener. So what is the minimum kind of limit above the background that we would consider to be a reliable detection number?
For microwatts, the limit of quantification. So we are working with an 0.5 microwatts, so 500 nanowatts.
Great. Okay, so what if we omit DMSO by harvesting from the plate? Do we still get detection delay issues?
No, if you just run without cryopreservation, then there's no issues. Then it will be more like your cut cells only. It will look more like this plot.
Great. So if the run lasts 3 days, is it possible to add more samples or free spots in the rack, or would that give too much disturbance?
So we're working to enable that. We call it random access. As it is now, is a batch insertion, but we are working to enable very quickly or soon that you are able to do several loads on the same plate, because you don't always have enough samples to fill the 32 positions, right? So yes, we want to be able to enable retracting the plate, loading new samples, removing positive samples that come within 24 hours, and make a more dynamic system. Absolutely. And that will be a software solution. Yeah.
Thank you. Is identification also possible when you have more than one species as a contaminant?
It depends on the combination of species. In our experience, it could be. It complicates things a little bit more, but it could be. I don't want to be too committed to that answer, because the possibilities are endless, right? Depends a lot on what are the species that you in the polymicrobial composition of the sample.
Are we using any additional reagents here, or is all the data that you've just shown with TSB, FTM, growth media directly on sample without filtering?
Yeah, no filtering, is direct inoculation. We are also working, although this will take a little bit more time, on a filtering solution so we can increase the volume. Yeah, but I forgot to mention that calorimetry is a completely blind method, so you don't have to add any dyes or any other chemicals or indicators. You just add the product and the media directly.
Great. How large cell densities have we tested with different types of product?
Yes, so typically we've been working with 1 million T cells per mil, but we have gone up to 100 million of cells. There is a higher background signal, but that is not a problem for detecting. So between 1 million and 100 million cells per mil we have tested.
Great. Now we move on to a bit of a regulatory question. And it's a good one. It's a big one. So just let me get into it and try and paraphrase a little bit. So, so we're claiming compliance with lots of different things. So what are the key regulatory steps now in terms of validation procedures required to transition from the traditional methods that we have over to a new technology like we're presenting here?
The key steps.
Yes, and if I can elaborate on that a little bit more, even kind of what are the specific examples? I don't know what examples exactly, maybe equivalency examples, data examples, to show in terms of validation that our method is actually superior to traditional methods.
Well, the points are, as I say, for qualitative methods are these points here, right? We need to show the sensitivity, the specificity, how robust and rugged the system is, what are the risks with it, and then of course the equivalency to compendial method, We need to show that we are not worse than the compendial, which we're in the process of doing. So hopefully we'll have that primary validation package now in September. Would that answer the question? I'm not sure.
If it doesn't, please write another question. Yeah. And we could potentially discuss regulatory aspects in more detail, perhaps in a separate call. We're definitely open to that. I think you've actually answered this in several questions here, but maybe more explicitly, what's the ratio of microorganisms and background cells that we typically test?
Well, now we'll have to do calculations, but there's 250 microliters of one. There's 250,000 T cells per 5 microbial cells. What ratio is that? Very low.
Exactly. I think we can...
So in one vial, you would have 250,000 T cells and maximum 5 CFUs, but it could be anything between 1 and 5.
Great. Okay, so is this an endpoint assay or continuous, so that we could enable continuous, kind of conditional release? Okay.
Yes, absolutely. It is continuous, and yeah, as you saw the thermograms, that's what you see. Exactly. Either is a downward slope, the equilibrates over time if it's a negative sample, or you get the positive sample, but it's continuous measurement.
Great. We have a question here about viable but non-culturable cells. Mm-hmm. So given that we can detect metabolic activity, yeah, what about these, you know, these of course represent big challenges for culture-based approaches. Do we have any data on that?
Yeah, well, not data, because unfortunately, if they are viable, so technically metabolically active, but they're usually so few cells that they fall under our limit of quantification. So if they're not actively growing and multiplying, it's a challenge for us as well.
Yeah. Does the workflow have to be performed within a biological isolator?
A biological safety cabinet, not necessarily an isolator. We work in biological safety cabinets only, and usually that works very well. Doesn't have to be isolator.
Perfect. I just had a comment here as well that viable non-cultural is the same problem with compendial sterility methods as well. Okay, this is the last question as I see it. If you have any more final questions to throw at us, please do so. Otherwise, maybe think about them later on, write them down, send them to us in an email. Thank you very much there, Marta, with your email address on. We would absolutely love to hear from you. We're extremely excited about our offering for the rapid sterility testing and cell and gene therapy markets. And we are extremely happy to have all of you with us today. So without further ado, I'd like to thank you, Marta, for the great presentation. And I'd like to thank everyone here for listening, and please look out in your inbox for the webinar replay. And please, please, please do feel free to contact us. So from us here at Symcel in Stockholm, thank you very, very much. Goodbye.
Thank you.